Before you begin working in the lab, it is important that you wash your hands before putting on your gloves. Once you have on your gloves, sterilize your work environment with either a 10% bleach solution or 70% ethanol solution. Spray down the entire area that you will be working on. The third transfer you will be making is from bacteria growing on an agar plate into sterile culture broth. When making this transfer, it is very important that when you transfer the bacteria from the plate into the broth, that you're only selecting a single isolated colony. You do not want to select from the lawn, as these could be mixed cultures and not pure. So again, make sure you are selecting a single isolated colony when making your transfer. Before beginning, look at your agar plate and make sure you see no visible contamination. Look at your stale broth and make sure that you see no contamination, as this should be sterile. Begin by making your label for your broth. As we reuse the glass tubes, please write on tape and not directly on the tube. Your label should contain your name, date, media type, and bacteria. When working with plates, always have the plate lying flat on the table. When opening the plate, you'll want to use the clamshell method. This allows you to use the lid as a shield, protecting the media and culture from contamination in the air. Always work near a Bunsen burner, the flame of the Bunsen burner, again, because this creates a convection current causing the air to rise, reducing the amount of bacteria likely to fall into your plate. Start by sterilizing your inoculating loop. Again, you will move slowly through the flame, starting from the base to the tip, making sure that each part of the loop turns red hot. Before inserting your inoculating loop into the plate, make sure that it is cooled. This will take 15 seconds. If you're unsure if your inoculating loop is cooled enough, you can always touch it against part of the plate that does not have any bacteria. If you hear a sizzle or see the agar melt, then your inoculating loop is too hot. To transfer bacteria on the plate into your broth, again, you will only touch the loop to a single colony of bacteria. Open the plate in a clamshell method and have the opening away from you also, so that when you breathe, you do not breathe into the plate. Put your loop onto a single colony. Grab your culture broth, open it again using the loop in your dominant hand like a finger, and using your pinky to open the cap. Now that it's open, always keep the cap facing down in your hand and always keep the broth open at an angle. Quickly pass the broth through the flame 2 to 3 times. Insert the loop. Remove the loop, pass the opening through the flame 2 to 3 times, close the tube, and place the tube upright on the tube rack. sterilize your knot getting loop. Once finished with your transfers, make sure to sterilize your work area. Turn off your Bunsen burner. And replace all bacteria to the appropriate designated areas. Before leaving lab, you will remove your gloves and wash your hands. It's important to wash your hands after removing your gloves.